tell you is a little different. And it's going to challenge the way you think about how you treat head and neck cancer. And so what I want you to do is just to kind of pull back for a second. I'm going to first of all thank the people who did the work. And some of them are in the audience today. So I have a young KO8 awardee, Dr. Spanos, who has done some of the work I'll talk about. Key people in the lab are Dan Vermeer, Paola Vermeer, and several of the students, Joe Kopic. The work done on metabolism is with another lab, the Miskimans lab. And what I want you to think about is two questions, and that is, so how does cisplatin radiation truly clear the tumor? Okay? And the second is, what things can we change safely in that environment and not impact survival? Okay? And those are the two questions I'm going to try to answer with this talk. So everybody's talked about the improvement or not improvement, and they mentioned it some on what has made this difference in survival. And anytime you have a dichotomy in science, it's really nice because you have two different, exact the same, histologically the same cancers, and yet there's a very different response. So we propose, just like I think Dr. Brazell mentioned earlier, that maybe we just got lucky, right? Over the time we've changed, and things, we just got lucky. We happen to be choosing the right radiation and chemotherapy. They're just more sensitive, and could explain the improved survival. And that great hypothesis, actually, that's what we thought too. And we actually tested it. And again, this is in cell lines, and you can choose different cell lines in sensitivity, but overall, we looked at several cell lines. And actually, as you increase the dose of radiation, which is shown on the bottom, you actually have a much more resistant line in your HPV positive head and neck cancer cell lines. And so we were surprised by that. This is also true for response to cisplatin. So we went on to further ask this in vivo, because in vitro stuff is very misleading. I can show you multiple levels on that. We made a mouse line. We cultured tonsil keratinocytes from a mouse. We transformed with the E67 oncogene, and very fortunately, they transform in an almost nearly identical way as humans do. And they grow in mice, and you can actually test that hypothesis a little differently. And again, these, we transformed the HPV negatives, and it's similarly knocked out common pathways of the E67 target, which Sherry did a nice job mentioning earlier. And again, these were more sensitive to both. I'm going to just show you the radiation, but the cisplatin, this has been published. And the HPV negatives were more sensitive. So we took that exact same line, and we put it in, the nice thing you do with a mouse in an immune-competent matter, and we put it in an immune-competent mouse. And the wild type is listed on the left. And a rag mouse has only innate immune response and not an ability to develop an adaptive T-cell response. And again, you see a dose response in both. However, we got long-term cures in the wild type. We then went on and did, in the paper, I'm not going to show you this, but we went on and did adoptive transfers, and you can actually restore the deficiency in rag by adoptively transferring it back into the rags. And so you can prove beyond a shadow of a doubt that radiation actually causes an immune response, and it's completely dependent to have an immune response to clear these tumors. It's very important as you think through your patients that you see and that I still treat as far as immune-suppressed patients. So the second thing we did is we made it HPV negative, and interestingly, you get a dose response in both the rag and the wild type, but you don't get clearance in this non-antigenic model. We went on and did the exact same thing for cisplatin. I'm going to show you this a little bit later. And interestingly, the cisplatin responses, again, completely to get a long-term cure of cisplatin, it's completely dependent on an adaptive immune response. I'm going to show you a little bit of that work. I don't have time to go through all that paper, but just to summarize that. So both cisplatin and radiation require an immune response, and that response is completely required for a long-term cure. And that's what's summarized in this slide. Sorry. Okay, so since we knew this is a complex environment, and I'm showing you it here. And so we went through and systematically thought in the lab. We said, okay, what is there, and what could be impacting this environment in order to cause this clearance or not cause this clearance, to make it better or worse? And I have a list of all, some of them, probably not every cell line that's present in that tonsillar epithelium. And so we first, since my lab focused on transformation, we first looked at the epithelial cell. Before we get into that, though, I'm going to go through a little bit, because somebody asked me earlier in one of the talks about, so what is required? And I'm going to go through these pretty quickly. So, and there's several different ways you can look at this. If you deplete CD4s with an antibody, actually, you get worse survival and no clearance. So again, it says what our RAG model did. Interestingly, you deplete NK cells. There were some talks earlier on this by the Klusman group, showed that you actually get a slight improvement. So it could be a regulatory component. This is going to come up a little later. If you just knock out one component in a CD40 receptor knockout, it's just like the RAG mouse. So the CD40 signaling during the adaptive response is completely required. CTLA-4 inhibition enhances survival by long-term survival by about 20%. And CD137, which is an activator, enhances survival by about the same, 20%. And I'm not going to go into each one of them. I'm going to show you examples a little bit in the end. So some of these things are already in therapy and being tested, but they do have activity in this immune-competent mouse model. But I'm going to get a little bit back to, because I think it ties into our thinking on these de-escalation therapies and also how we think about the current, how cisplatin radiation work. So one of the things you have is we hypothesize that, again, the epithelial cell is here and it has to be sensed. It can be sensed by direct contact. There's these things called exosomes that can be shed off. But we hypothesize that the change would be at the cell surface. And that what's being sensed is then either touched on with a dendritic cell, or it's sent off and that dendritic cell senses it through these things called exosomes. And the first thing we looked at is, again, very simple things to do in the lab. You can either say, okay, here's a cell we radiated, or here's a cell we didn't radiate, and what changed, and does that impact survival? So we looked at several things that are called DAMPs, or they're things that as a cell becomes injured, it changes the immune response. And some of these have been well described, like calreticulin did go up, and that is an immune stimulant. But the one I want to focus on is CD47. This is a response that if you look, with radiation, you see a dependent loss. And it's not, this is a control showing that it's on the cell surface. These are the internal cells. These were biotin labeled. Some of the things that had been reported, we did not find a change with radiation in this model. So again, this is a don't eat me signal. So, and it's overexpressed in HPV positive head and neck cancers and actually HPV negative head and neck cancers. And so as it interacts with antigen presenting cell, it triggers a more of a tolerance type. And it happens both in all of our cell lines that we tested. These are the responses in our HPV positive, HPV negative cell lines, as well as our mouse cell lines. It's dose dependent. And so with a higher dose, and if you accumulate your doses, you see the same thing as the dose dependency shown here. And actually, you lose it from the cell surface, and that's shown here. This is a control cell. It's completely studs on a confocal cell surface. After they radiate, they get kind of in these punctate little vesicles. The same thing happens. This is actually in vivo, and we do like to look in vivo. So this is the CD47 of the tumor. This is muscle and skin. At 24 hours, it completely goes away. It starts to slightly come back at 48 hours, but again, you see, and it does slightly arrive at 5. Unfortunately, this picture didn't show up, but it does slightly come back. So it's if you take enough time off, the CD47 signaling does return, and you see that also in vitro. And this just shows the interaction with the dendritic cells. We took dendritic cells and mixed them with green cells, and then you can either irradiate them, and you can see a great increase on flow when you radiate, or you can block the CD47, which again would allow more uptake. And you see, compared to its control antibody, a significant amount of uptake into the dendritic cell. But this is what I like. We actually do these experiments first, and then we try to figure out the mechanism after, because I get bored otherwise in the lab. And so we depleted CD47 compared to a scrambled control. And then we chemoradiated. And you can see these are individual mouse curves on the left. But you can actually see an improvement in 50% survival if you get rid of the CD47 from the cell surface. And this is an immune-competent model. And you see the exact opposite in immune-incompetent model. Actually, the CD47 depleted ones grow faster, proving that it's not actually It's not actually a change in the way the cell grows or sensitivity to radiation that allows the sensitivity to occur. And I'm not going to bore you guys because my eyes start glazing back when I hear people talking about all the basic mechanism too. But I'm going to tell you that the same thing happens in a little bit different manner with cisplatin. I'm going to shorten the story. So as you increase your dose of physiologic doses of cisplatin, a compound called CD40 ligand, which binds to the CD40 receptor, actually starts increasing. And this is shown nicely in this kind of dose-dependent response in our HPV positive, HPV negative cell lines. This kind of shows it more in both our mice in a time and dose-dependent manner, as well as our HPV positive and HPV negative cell lines from human. The interesting thing, and this is a really cool molecule, it's the only thing that's shown as a single agent, complete long-term cures in our mice. And the two comparisons you need to look at are the blue line, is just an IgG isotype control. And if you give just single doses of CD40 ligand, actually you get about a 20-25% long-term cure rate. And these are big tumors when we start these mice. If you do it in conjunction with chemo radiation, interestingly, you improve survival by about 40%. Long-term survival, these mice are cured of their cancers by about 40%. So I'm going to just summarize it, and these are not all the changes, I will guarantee it, that go on, and it's a complex environment. But from epithelial cell, we at least know a few things. The synergy happens undoubtedly on direct cell toxicity when we think through these trials. But it also happens on the immunologic signaling standpoint. And you have to consider that both when you're treating patients who are immune depleted, like I've actually taken 2 transplant patients off of their HPV positive patients off of their immune suppression, and they were cured because the 2 before it both died of their disease. And so I think it becomes a very important factor clinically when you're treating patients. Okay, the next part of the story I'm going to tell you is, this is also a complex, and you guys all know this. So what's PET positivity based on? FDG. Correct. And I'm going to build an FDG uptake, and the reason why it is because they synthesize ATP about 18-fold less efficiently. And what I'm going to build an argument is that environment creates a lactate environment, and that changes your, even if you have it correctly, that changes your ability to have an immune response if it's there. And so it's been known for a long time. So we had this response, and actually some of the mice don't clear. We went back to the literature and we looked, okay, what's been completely associated with poor prognosis to chemo and radiation or both. And interesting, lactate production has been shown time and time again. I'm going to build that. And interestingly, if you take there's a lot of literature now on antigen-presenting cells in the tumor microenvironment versus the periphery. And if you take tumor cells and you put them in lactate, they become tolerant. And they express a very different pathway than those that have not been in that environment. And that's what's shown here. That in lactate, it's dependent on lactate. And the other thing we know from us treating abscesses is that even if you have the wrong environment, you need your CD8 cells to work. And both perforin and granzyme are completely inhibited in the presence of tumor levels of lactate. So even if you have a response, is it effective? The reason I bring this slide up is because I go back to chemistry always, and I always was really nervous about where the CO2s came off when I took that test. And but really, when we think about this, there's targetable mechanisms, and these things self-target to the tumor, unless you're like me running on a run where I probably use a lot of anaerobic glycolysis because I'm not in good enough shape. But our normal body does not utilize this mechanism at all under normal physiologic conditions. And the reason why our squames do it so much is because they don't have oxygen. And so you can give it to the body, and the rest of the tumor cells do not, the rest of the normal cells will not be affected by these type of class of chemicals. And so the ones I'm going to talk about today are a little bit are metformin, which poisons the step 1 of the coenzyme Q pathway. And the other one is DCA, which blocks PDK1, allowing entry and blocking entry up toward lactate production. And these things do change lactate production. Metformin at physiologic doses in our cells in vitro greatly increases lactate production. And DCA inhibits it, not quite as dramatically as some other compounds, but that's what's shown here. This is actually what I like to look at, and these are the in vivo measurements. You can actually measure these with electrodes in vivo. You can also cut the tumor out and immediately flash freeze it. You can get a quantitative measure of lactate production. And this just shows you can statistically regulate at physiologic doses the amount that a tumor produces lactate in vivo. Sorry. So we did the exact same spread I showed you now multiple times in our mice. So we actually did this experiment. We gave the mice physiologic doses of metformin, did chemoradiation compared to a control injection, or we gave them DCA. And we improved long-term survival with DCA by about 25% consistently in our mice. Metformin almost makes it like a rag mouse. You lose a lot of survival with the addition of metformin during chemoradiation. And you can rescue that response by the addition of DCA, so you can actually rescue that decrease in survival. What's shown on the right is that RAG1s, again, it's an immune-dependent response, so none of them ultimately survive. So we've actually finished a phase 2 trial in 50 patients using DCA in this setting, and I will tell you, it just finished in June. We're unmasking it in August. Is that I can't tell which ones got DCA and which ones didn't, as far as from a toxicity standpoint. And I don't know how efficacious it's going to be, but these patients tolerated this. It doesn't. So it's not like a lot of the other compounds that add a lot of morbidity. These agents that are going to be coming on the market are going to be very well tolerated, no matter what setting you put them in, because again, they self-target. And we don't have, there are very few side effects, even if you read the DCA literature, to taking DCA long-term, and it's been done in these patients who have inborn errors of metabolism. Last thing, this is going to be very quick, because this is work that Sarah Pei has kind of pioneered, frankly. We've looked at other agents in what I would consider a safe de-escalation study. And so what we looked at is the PD-1 inhibitor. Merck gave us the compound. I have no connection with Merck. But we did it as a single agent or in conjunction with standard chemoradiation. And this is the results. So as a single agent, it has no activity at all. There's absolutely no difference in growth. But this is what's cool. So you can improve this agent in conjunction with the stimulation that you see with chemoradiation and improve survival by about 57%. And these are 12 or 13 mice. And we didn't see, I will tell you with the CD40 activation, we saw some toxicity. We do not see a lot of increased toxicity in mice. Now, they're a little bit harder to judge in humans as far as autoimmunity. But again, a very well-tolerated regimen. And at least in a mouse, fairly safe. And I know that these are going in clinical trial in our patients in multiple levels. So if you guys do hear it, there's a great trial. I gave the talk at ASCO before Thai, and it's amazing. These patients, I mean, if you look at the response rates as a single agent, it's for about 5 to 10% of the patients. It's the only thing I've seen where their tumor melted away and they're cured. And so it's a cool therapy. I think we need to understand where to put it, how to get it in there, but that's what's coming down the line for new therapies in head and neck. So to conclude, I'm just going to say that, again, think through the things, understand mechanism of why things happen. It's very important when we think about how to add or how to take away, and that the immune response is incredibly important in clearing probably both HPV positives and HPV negatives, frankly, based on the PD-1 data. Second is that modulating certain components is going to be very safe, and in the future, not very distant future, capable. So thank you, and I'll take any questions.
John.
Yeah.
Very elegant work as usual. The relationship of the PD-1 inhibition and the chemoradiotherapy, two things. One, do you think it matters when you give the PD-1 in conjunction with the chemoradiotherapy? And did you do that study? And I guess secondly, what is the mechanism? Is it the release of immune stimulants by the chemoradiotherapy, that then helps the PD-1 to work because it's not as effective as a monotherapy?
Two great questions. So the first one is timing, and we have just started these experiments. I will say you can see a little bit from timing. If you don't give any chemoradiation, the timing doesn't matter. You know, again, we let the tumors get very large. They're probably about 1 centimeter, and so if you do that, that's about a 6, 8, 10 centimeter lymph node if you look at it in us. And so, do I think timing matters? Yeah, and when to do it, and when to do it safely. Again, when we start looking at a T cell response, it typically takes a couple weeks. So we wanted to kind of get rid of the inhibitory response, and that was our rationale for starting it a day before we did the chemoradiation, so that we're going to stimulate the response and get rid of the breaks. And again, I think that might be the right one. How long the stimulus lasts during therapy, I would love to know the answer to that question. We do see a lot of regression and inflammation early on. And so I think it happens pretty quickly. So I guess my gut feeling would be I would start it early in your therapy if you're thinking about moving into clinical trial. And I don't think the chemoradiation is the only way to induce this. And there might be a lot safer ways. So again, we start thinking completely outside the box. I actually think, again, I'm a surgeon. I'd love to be able to give a little immunotherapy, have it nearly completely regress, and just take a little lymph node out of the neck, you know? And I think as far as long-term toxicity, I know it might leave Dr. Brazel completely out of business, but again, it's doing what's right for our patients. I am committed to the least amount of morbidity with the longest cure, and so I think those are the things that we need to challenge ourselves with in the future.
Yeah, I'm a surgeon. I'm a surgeon too. Why don't you want to put them out of business?
You guys don't scare me.
Well, thanks everybody, and thanks for your attention.
Well, I'd like to thank all of our speakers in this panel today, and I think we're finishing on time. Thank you.